500 million years ago, the land was silent. Not the silence of an empty room or a quiet morning, and something far more absolute. A silence without memory, without interruption, without the possibility of sound at all. No wind moved through forests because there were no forests. No creature called out across open ground because nothing with a voice had ever stood on it. The continents existed as pure geology, vast slabs of rock baking under a sun that offered no mercy, swept by ultraviolet radiation so intense it would have dissolved living tissue within minutes of exposure. This was not a world waiting to be discovered. It was a world actively hostile to the idea of being discovered. Look at those ancient continents, and what you see is almost architectural in its starkness. Erosion had sculpted shorelines and river deltas, but there was no color beyond gray and rust and the pale ochre of oxidized minerals. No roots had broken stone into soil. No organism had yet begun the slow generational work of transforming dead rock into something that could sustain life. The closest thing to vegetation was a thin, almost invisible film of microbial life. Cyanobacteria and early algae clinging to wet surfaces near the water's edge, forming mats of biological material so delicate they would leave almost no trace in the fossil record. They were there, doing something profound, quietly exhaling oxygen into an atmosphere still sorting itself out. But they were not yet the foundation of anything we would recognize as an ecosystem. They were pioneers in the truest sense, alive in a place that had not yet learned how to support life. Meanwhile, beneath the surface of the ocean, an entirely different world was operating at full complexity. The seas of the late Cambrian and early Ordovician were not primitive or sparse. They were extraordinary. Reefs and seafloors teemed with organisms of bewildering variety, trilobites navigating the substrate with compound eyes, filter feeders suspending themselves in currents, predatory creatures that had already refined the mechanics of pursuit and evasion across hundreds of millions of years of evolution. The ocean had become life's masterwork. It offered chemical stability, thermal buffering, buoyancy that relieved the body of gravitational stress, and an ancient, reliable infrastructure of nutrients and dissolved gases. Everything an organism needed to survive had already been solved there. The ocean was not just a habitat, it was a kind of perfection, carefully assembled over geological time, and life had shaped itself entirely around it. This is what makes the transition to land so difficult to explain with simple logic. There was no obvious reason to leave. The ocean was not failing. Resources were not depleted to the point of crisis. There was no single catastrophe pushing life toward the shore, and yet something began moving in that direction. Not with intention, not with foresight, but through the relentless arithmetic of evolutionary pressure working on populations across thousands of generations. The question that sits at the center of one of biology's most consequential stories is not simply how life moved onto land, but why the conditions of survival eventually made the impossible option worth attempting. Part of the answer lies in the shoreline itself. The boundary between ocean and land during this period was not a clean, stable line. Tidal zones were dynamic and unpredictable, areas where the sea advanced and retreated, where pools of water formed and evaporated, where salinity fluctuated and oxygen levels swung dramatically depending on temperature and time of day. These environments were by any measure more difficult than open water. And yet difficulty, in evolutionary terms, is not always a deterrent. Difficulty is sometimes a door. Organisms that could tolerate the stress of a tidal pool, surviving brief periods of low oxygen, enduring changes in water chemistry, managing the physical turbulence of waves, were organisms that had already begun developing capabilities beyond what the stable deep required. They were being shaped by a more demanding curriculum. The oxygen story alone carries extraordinary weight. Atmospheric oxygen levels during this period were still significantly lower than what exists today, and the distribution of oxygen in shallow, warm, fluctuating water was inconsistent in ways that deeper marine environments were not. Creatures caught in tidal pools at low tide faced the very real possibility of hypoxia, insufficient oxygen to sustain normal metabolism. Those that survived were not simply lucky. They were, in some cases, organisms whose anatomy allowed for supplementary gas exchange, whose physiology could slow down under stress, whose behavior drew them toward surfaces or air pockets. These were not adaptations built for land. They were adaptations built for survival in water. But they were also, in retrospect, the first architecture of a creature that could one day breathe air. There is something almost unbearable in the slowness of what was happening. No individual organism experienced a transition. No single creature decided to cross a boundary. What was occurring was statistical and incremental. Population shifting, traits accumulating across lineages so gradually that no single generation would have recognized the change taking place within itself. And yet, across millions of years, the shoreline was becoming a laboratory. Each tidal cycle was a filter. Each drought and flood was a selection event. The creatures that remained in those marginal zones were not the same as the creatures that had always lived safely in open water. They were something subtly different, something being rewritten. And in the shallows, where warm water moved over ancient rock, and the tide pulled back to expose unfamiliar surfaces to unfamiliar air, something was already moving that would one day change the shape of every continent on Earth. It had not yet taken a step, but it was already experimenting with the question of what a step might mean. The animals did not arrive first. That is perhaps the most underappreciated fact in the entire history of life's conquest of land. That by the time the first creature with legs or fins or any recognizable form of locomotion pulled itself onto solid ground, the work of making that ground survivable had already been underway for hundreds of millions of years. The land was not taken by force. It was prepared. Slowly, invisibly, through biological processes so gradual and so small that no individual organism participating in them could have registered what was being built. Before there were footsteps, there was a transformation, and it began not with animals, but with something far older and far less celebrated. Cyanobacteria are among the oldest living organisms on Earth, their lineage stretching back more than 3 billion years into the planet's early history. For most of that time, they lived in water, ancient seas and shallow lakes, forming layered mats called stromatolites that are still visible in certain parts of the world today, as fossil records of a biological revolution most people have never heard of. What cyanobacteria did quietly and without any mechanism beyond their own metabolic chemistry was begin the process of filling Earth's atmosphere with oxygen. They performed photosynthesis. They split water molecules, released oxygen as a byproduct, and over geological timescales so vast they resist genuine comprehension, they altered the chemical composition of the air itself. The Great Oxidation Event, occurring roughly 2.4 billion years ago, was not caused by a geological upheaval or a cosmic collision. It was caused by biology operating at a microbial scale across an incomprehensible span of time. Cyanobacteria did not intend to remake the world. They simply lived, and the world was remade around them. By the time we reach the Ordovician period, roughly 470 million years ago, this oxygen legacy had become the foundation upon which everything else would be built. Atmospheric oxygen had risen to levels sufficient to begin forming a meaningful ozone layer. The ultraviolet shield that land had previously lacked entirely. This was not a minor development. It was the unlocking of a door that had been sealed since the planet formed. The land surface, previously bathed in radiation lethal to complex organic molecules, was becoming incrementally less hostile. Not hospitable, not yet, but less actively lethal. The chemistry of survival was slowly, measurably shifting in favor of the possible. Into this altered world came the first terrestrial pioneers that most biology textbooks treat as footnotes. The early non-vascular plants, or more precisely, the organisms that gave rise to them. Genetic and fossil evidence now converges on the conclusion that land plants descended from a group of freshwater green algae called charophytes, organisms that already possessed several of the biochemical tools that land life would require, including the ability to manage desiccation stress, to protect their cellular machinery from ultraviolet damage, and to reproduce in environments where water was not always continuously available. Their transition to land was not a single event, but a gradual ecological expansion, a slow colonization of wet margins, riverbanks, lake shores, and coastal flats where moisture was reliable enough to sustain early biological experiments. The earliest land plants left an extremely sparse fossil record, in part because they lacked the hard, mineralized tissues that preserve well over geological time. But the molecular clock, the technique of measuring genetic divergence rates to estimate when evolutionary lineages separated, suggests that the first land plants appeared somewhere between 470 and 500 million years ago. They were almost certainly simple, low-growing organisms resembling modern liverworts, flat, lobed structures with no true roots, no vascular system for transporting water internally, no leaves in any recognizable sense. They spread horizontally across damp surfaces, absorbing moisture directly through their tissues, anchoring themselves with primitive root-like structures called rhizoids that were capable of penetrating the surface of rock and beginning the slow chemical work of weathering it. This weathering process deserves far more attention than it typically receives. When early plants sent rhizoids into rock, they were not merely seeking purchase. They were, through the release of organic acids and the physical pressure of biological growth, beginning to chemically dismantle the mineral structure of ancient stone. Silicate minerals were dissolved. Calcium and phosphorus were released into the surrounding environment. The rock surface was being transformed, particle by particle, into something that could hold moisture, accumulate organic material, and eventually support the growth of more complex biological communities. This was the beginning of soil, not the rich, layered, biologically active soil of a modern forest floor, but the first tentative steps toward a substrate capable of supporting life that did not need to cling to wet rock to survive. And then there were the fungi. The relationship between early land plants and fungal networks is one of the most important and least intuitive stories in evolutionary biology, and it almost certainly predates the plants themselves. Fungi are neither plants nor animals. They belong to their own kingdom. Their biology built around the decomposition and extraction of nutrients from organic and inorganic material through the secretion of digestive enzymes and the extension of thread-like structures called hyphae. When these hyphae penetrate rock and soil, they release acids that dissolve minerals with remarkable efficiency, making nutrients bioavailable that would otherwise remain locked inside stone for millions of years. Fossil evidence from the Ordovician period includes structures that appear to represent early mycorrhizal networks. Associations between fungal hyphae and the root-like structures of early plants in which the two organisms exchanged resources in a relationship of mutual dependence. This symbiosis is difficult to overstate in its significance. Early land plants lack the elaborate root systems that modern plants use to mine the soil for water and nutrients. In a landscape where true soil barely existed, their own biological capacity to extract resources from bare rock was limited. Fungal networks extended their reach enormously, threading into the mineral matrix of ancient stone and delivering phosphorus, nitrogen, and water directly to plant tissues in exchange for the sugars that photosynthesis produced. The plant could not have expanded as effectively without the fungus. The fungus gained a reliable source of carbon it could not produce itself. Together they were more capable than either was alone. And together they began the systematic transformation of barren rock into biologically active substrate across areas that no single organism could have colonized independently. What this meant for the land surface over millions of years was nothing less than planetary engineering at a biological scale. As early plants and their fungal partners spread across wet continental margins, they accelerated rock weathering at rates that purely geological processes could not match. The breakdown of silicate rocks through biological activity draws carbon dioxide out of the atmosphere as part of the chemical reaction, which means that the expansion of land plants during the Ordovician and into the Silurian period had a measurable effect on global carbon cycling. Atmospheric carbon dioxide levels fell. The planet cooled. Ice ages followed. One of the most severe glaciation events in Earth's history occurred near the end of the Ordovician, in part as a consequence of the carbon sequestration driven by biological expansion on land. Life was not simply adapting to the planet. It was changing the planet's chemistry in ways that would reshape the conditions for all future evolution. As organic material accumulated on the land surface, dead plant tissue, fungal residue, the chemical byproducts of biological weathering, the substrate began to develop something resembling structure. Water retention improved. Mineral nutrients became more concentrated in the upper layer of what was slowly becoming genuine soil. The surface that had previously offered nothing to any organism attempting to live on it was beginning to offer something specific and valuable: stability, resources, and a biological scaffolding upon which more complex communities could potentially establish themselves. The transformation was not uniform across the planet, and it was not fast by any human measure. But it was cumulative, and cumulativity, given enough time, becomes geological fact. By the time the Silurian period arrived roughly 440 million years ago, the land surface in many regions bore only a faint resemblance to the sterile rock of the Cambrian. Low carpets of non-vascular plants extended across moist areas near water. Fungal networks threaded through the developing soil layer. Microbial communities had diversified to exploit the new chemical environments created by biological weathering. Oxygen levels in the atmosphere continued to rise. The ozone layer continued to thicken, and the ultraviolet threat that had once made land surface exposure a death sentence was gradually becoming manageable for organisms with even modest protective biology. The land was not yet a forest. It was not yet a landscape that any modern eye would recognize as verdant. But it was no longer sterile. It was, in the most precise sense of the word, becoming habitable. The significance of this sequence, oxygen first, then soil, then the slow biological carpet of early plant life, is that it establishes something fundamental about how evolutionary opportunity actually works. Animals did not invade a hostile frontier through some exceptional act of biological courage or metabolic ingenuity alone. They moved into a territory that had been prepared for them across hundreds of millions of years of prior biological labor. The pioneers were invisible and their work was chemical. They asked for no recognition and left only the faintest fossil traces. But without them, the story of land-dwelling animals, every amphibian, every reptile, every mammal, every human being who has ever walked across solid ground, simply does not begin. The land had been transformed from the outside in, molecule by molecule, generation by generation. And somewhere along that prepared margin, in the thickening substrate and the oxygen-richer air, creatures that had never been anything but oceanic were beginning to encounter a question they had no framework to answer: whether the world that had been so carefully, so unknowingly built for them was finally worth entering. The first creature to leave a permanent record of its passage across solid ground did not know it was making history. It had no awareness of the ocean it had come from, no understanding of the boundary it was crossing, no sense that its movements through a thin film of coastal mud would eventually be read by organisms that would not exist for another 400 million years. It was simply moving, driven by chemistry and reflex and the ancient biological imperative to find food, avoid threat, and continue existing. But the mud held its impression. The stone preserved what the mud became, and what we now hold in our hands when we examine certain rock formations from the Cambrian and Ordovician periods of ancient continents like Gondwana and Laurentia are the oldest confirmed footprints on Earth. A series of paired, shallow impressions pressed into ancient sediment by something small, armored, and utterly unlike anything that had walked anywhere before. The trackways themselves are understated objects. They do not announce their significance. A series of small indentations arranged in bilateral symmetry across a slab of sedimentary rock. They could be mistaken by an untrained eye for nothing in particular, a pattern in stone with no obvious story attached. But paleontologists examining trackways like those found in the Cambrian deposits of Canada, or the slightly younger but equally remarkable traces discovered in Poland and across parts of the Arabian Peninsula, have recognized in that symmetry something precise and deliberate. The gait signature of an arthropod, moving with coordinated limb pairs across a wet substrate, pressing its weight evenly into the surface with each stride. The creature that made these marks was not swimming, it was not drifting, it was walking. And in the history of life on this planet, that distinction carries almost incomprehensible weight. To understand why arthropods were first, you have to understand what land actually demanded of any body that attempted to cross it. The problems were not subtle. Gravity, which is essentially neutralized in water through buoyancy, becomes a full and constant force on land, meaning every part of an organism must be structurally capable of bearing its own weight without the support of the medium surrounding it. Water loss through evaporation is continuous and potentially fatal for organisms whose biochemistry depends on high internal moisture levels. The uneven, unpredictable surface of rock and early soil substrate requires limbs capable of adjusting dynamically to terrain that does not offer the uniform resistance of water. And the respiratory challenge, extracting usable oxygen from air rather than from water, required either the rapid development of new anatomical structures or the fortunate possession of existing ones that could be repurposed. Arthropods as a group had already solved most of these problems before land even became a viable destination. The exoskeleton, the defining feature of the arthropod body plan, is a marvel of functional engineering that serves multiple purposes simultaneously. It provides structural support, replacing the hydrostatic pressure of water as the framework against which muscles can act. It resists compression and tension in ways that soft-bodied organisms cannot manage in a gravitational environment. And critically, it is composed of chitin, a biological polymer that is largely impermeable to water, meaning that an arthropod wearing its skeleton on the outside is an arthropod that loses moisture to evaporation far more slowly than any soft-bodied alternative. The exoskeleton did not evolve for land. It evolved in water, almost certainly as protection against predation. But in the transition to terrestrial existence, it turned out to be precisely the right tool for a problem that had not yet been formulated when the tool was made. The jointed limbs were equally fortuitous. In water, arthropod limbs function as paddles, fans, and grasping appendages across a wide range of forms. But the jointed architecture, multiple rigid segments connected by flexible junctions capable of moving independently, translates with remarkable efficiency to locomotion across irregular solid surfaces. A limb that can extend, flex, and rotate at multiple points along its length can adapt its contact angle to the specific geometry of whatever surface it encounters. Early land surfaces were not smooth. They were fractured rock faces, uneven sediment plains, crusted microbial mats, and the variable terrain produced by erosion and tidal action. A creature that could adjust each limb independently in real time to the microtopography beneath it had an immediate and decisive mechanical advantage over anything that could not. Size in this context was another quiet advantage. The earliest terrestrial arthropods were small, many of them comparable in scale to modern woodlice or centipedes, with body lengths measured in centimeters rather than anything larger. Small bodies mean low absolute mass, which means the gravitational force that must be managed is proportionally manageable. A creature weighing fractions of a gram experiences gravity as a far less demanding engineering problem than a creature weighing kilograms. The exoskeleton, which works efficiently at small scales but becomes increasingly problematic at larger ones due to the physics of surface area to volume ratios, was ideally suited to the size range of these early pioneers. They were, in a meaningful sense, the right size for the problem they were solving. Among the earliest confirmed terrestrial arthropods were organisms resembling modern millipedes, multi-segmented detritivores whose bodies were organized around the consumption of decaying organic material. The ecological logic here is worth dwelling on. Land, by the time these creatures arrived in the Silurian period, was not entirely without food. The biological preparation described by the spread of early plants and fungal networks had created a surface layer of organic debris, dead plant matter, fungal residue, the accumulated chemical byproducts of millions of years of biological weathering. A detritivore does not need to hunt. It does not need to pursue mobile prey, assess threats from other predators, or navigate complex social dynamics. It needs only to move through substrate that contains digestible organic material, which the early land surface in its damp margins near water was beginning to provide. The first terrestrial arthropods were, in the most ecologically coherent way possible, perfectly matched to the food source that the plant pioneers had inadvertently created for them. Respiratory adaptation in these early terrestrial arthropods is a subject that has generated considerable scientific discussion because the fossil record does not always preserve soft tissue structures with the fidelity needed to reconstruct internal anatomy with certainty. What we can infer from the phylogenetic relationships between early terrestrial groups and their aquatic relatives is that at least some lineages possess structures capable of gas exchange in air that were derived from gill-like precursors modified through evolutionary time. Book lungs, the respiratory structures found in modern arachnids, consisting of layered sheets of vascular tissue arranged like the pages of a book within a protected internal chamber, are believed to represent one pathway by which ancestral gill structures became internalized and repurposed for atmospheric oxygen extraction. The internalization was critical. A respiratory surface exposed directly to dry air loses moisture through evaporation at a rate that would rapidly compromise function. By drawing the gas exchange surface inside the body, behind a small opening that could be partially controlled, early arachnids achieved a degree of respiratory efficiency in air that made sustained terrestrial activity physiologically viable rather than merely transient. The arachnids deserve particular attention in this story because their fossil record, while imperfect, places them among the earliest confirmed terrestrial animals with certainty. Scorpion-like organisms appear in Silurian deposits with anatomical features consistent with air breathing and fully terrestrial locomotion. Whether the earliest arachnids were genuinely terrestrial or were instead semi-aquatic organisms spending time in both environments is a question the fossil record has not fully resolved, but the trajectory is clear. The arachnid body plan, with its internal respiratory structures, its highly impermeable cuticle, its compact body organization, and efficient locomotory architecture, was one of the first successful templates for permanent life on land. Now reconstruct the moment, not with the abstraction of geological timescales, but with the specificity of a single afternoon, or what would have been an afternoon, on a Silurian shoreline 430 million years ago. The sun is still higher in the sky than we would expect, the atmosphere slightly different in its composition, the light filtering through air with less particulate matter and fewer complex atmospheric interactions than modern skies produce. The shoreline is a place of contrast. Beyond the narrow band of damp sediment and shallow water, the land extends in colors of gray and pale green. The green supplied not by grasses or trees, neither exists yet, but by the low spreading carpets of early non-vascular plants and the dark filamentous networks of fungal colonies threading through the upper surface of ancient soil. The air is not silent anymore because wind exists, and wind across bare rock and early plant communities produces sound, though nothing like the layered acoustic texture of a modern landscape. Onto this surface moves something small. Its body is segmented, its surface chitinous and darkened, its many legs operating in coordinated waves of rhythmic motion that propel it forward with a mechanical efficiency that is almost mesmerizing in its precision. It has moved out of the shallow water margin, perhaps following the accumulation of decomposing plant material that has drifted above the tide line, perhaps responding to the sensory input of chemical gradients in the damp sediment that indicate food. It does not hesitate at the water's edge in any meaningful sense. There is no deliberation occurring. There is only the continuous operation of a nervous system processing environmental information and generating locomotory output in response. But each of its legs is pressing into the sediment as it moves. Each contact point is leaving a shallow impression in the wet surface. It is without any awareness of the fact, writing the oldest signature of animal life on land. Behind it, the impressions fill slowly with water and sediment. Most will not survive. Most will be erased by the next tide, by rain, by the geological processes that recycle surface material back into the substrate continuously and without sentiment. But some will not be erased. Some will be buried under sediment rapidly enough that the impression is preserved before it can be destroyed, compressed over millions of years into rock, and eventually lifted by tectonic activity back toward the surface where, after an interval of time so long it defeats any attempt at emotional comprehension, another creature, bipedal, large-brained, endlessly curious about its own origins, will find it and understand what it means. The land was no longer quiet. Something was moving across it. Something small, something armored, something that had emerged from the water not as a dramatic act of evolutionary ambition, but as the incremental consequence of biological pressures accumulating across millions of years of preparation. It did not know what it was starting, but what it was starting was everything. The ocean had given vertebrates everything. It had given them the neural architecture of predator and prey, the hydrodynamic body plans refined across hundreds of millions of years of aquatic competition, the sensory systems capable of reading pressure waves and chemical gradients through water with extraordinary precision. It had given them bone, which first appeared not as a structural support for movement on land, but as a mineral reservoir and a protective sheath around neural tissue in creatures that never left the sea. The vertebrate body, by the time the Devonian period arrived, roughly 419 million years ago, was a masterpiece of aquatic engineering. Flexible, efficient, metabolically sophisticated, and organized entirely around the physics of movement through a dense liquid medium. It was also in the shallow swamp ecosystems beginning to spread across low-lying continental margins, increasingly inadequate for the conditions it was being asked to survive. The Devonian world was dramatically different from the barren, ocean-dominated planet of the Cambrian. Land plants had diversified spectacularly. The first true forests were appearing, not the towering canopied structures of later geological periods, but genuine stands of woody vegetation several meters high, their root systems penetrating deeply into developing soil layers and fundamentally altering the hydrology of continental surfaces. Rivers and deltas spread across lowland terrain, fed by the runoff from these increasingly vegetated landmasses. And in the warm, low-lying basins where river systems slowed and spread across flat terrain, an entirely new kind of ecosystem was forming, the shallow swamp. Dense with organic material, stratified in temperature, and subject to seasonal fluctuations that could reduce expanses of open water to isolated, shrinking pools. These swamp environments were productive and biologically rich in ways that open ocean could not replicate. They were also, beneath their apparently hospitable surface, crucibles of physiological stress. The critical problem in a shallow, warm, organically dense body of water is oxygen. Decomposing organic material consumes dissolved oxygen as it breaks down. driven by microbial activity that accelerates dramatically in warm temperatures. In a deep, well-mixed body of water, this depletion is offset by circulation and gas exchange at the surface. In a shallow pool or a sluggish swamp channel with limited circulation, dissolved oxygen levels can fall to concentrations that most fish physiology cannot tolerate. The technical term is hypoxia, insufficient oxygen in the water to sustain aerobic metabolism through gill respiration. And in the Devonian swamp ecosystems, it was not an occasional crisis event. It was a recurring, seasonally predictable feature of the environment that any organism hoping to survive there had to somehow manage. Lobe-finned fish, the Sarcopterygians, whose living representatives include the coelacanth and the lungfishes, were already anatomically unusual among fish in ways that would prove to have enormous consequences. Unlike the ray-finned fish that dominate modern aquatic ecosystems, lobe-finned fish possessed fins supported not by rays of bone radiating from a simple base, but by a muscular, fleshy lobe containing a core of bones arranged in a proximal to distal series. One bone, then two bones, then a cluster of smaller bones toward the fin's edge. The correspondence between this arrangement and the structure of the tetrapod limb, one upper bone, two lower bones, a cluster of wrist or ankle elements, and then digits, was identified by 19th century comparative anatomists and has since been confirmed and elaborated by molecular developmental biology in ways that leave no doubt about the homology. The lobe fin was not a limb, but it was built on the same architectural blueprint, and that blueprint, under the right conditions, could be modified into something that functioned in ways fins had never functioned before. Some lineages of lobe-finned fish had also independently evolved structures capable of gas exchange using air rather than water. The relationship between the swim bladder of modern fish and the lungs of tetrapods has been debated by biologists for over a century. But the developmental and genetic evidence now strongly supports the conclusion that both structures are modifications of the same ancestral organ, a paired outgrowth of the digestive tract that could be filled with gas. In some lineages, this structure became primarily hydrostatic, used for buoyancy control. In others, those under consistent pressure from aquatic hypoxia, it retained and expanded its respiratory function, becoming a primitive lung capable of extracting oxygen from air gulped at the water's surface. Living lungfishes, the closest surviving relatives of the tetrapod lineage among fishes, still breathe air obligately in low oxygen conditions, surfacing with a regularity that reveals the ancient solution their ancestors developed to the problem of the Devonian swamp. This is the ecosystem into which Tiktaalik rosei was born, approximately 375 million years ago, in what is now the Canadian Arctic. A fact that itself requires a moment of geographical adjustment, since the Canadian Arctic during the late Devonian occupied a tropical latitude, its landscape a warm, low-gradient river delta system, of exactly the kind that drove the respiratory and locomotory experiments we are examining. Tiktaalik was discovered in 2004 by a research team led by Neil Shubin, who had predicted its existence and approximate age through an analysis of the evolutionary gap between fish and the earliest confirmed tetrapods, then deliberately searched the geological formations, most likely to preserve organisms from the transitional period. The prediction was vindicated with a completeness that remains one of the most compelling demonstrations of evolutionary theory's predictive power in the entire fossil record. What Tiktaalik had was a body that did not fit cleanly into any existing category. It had scales and gills, fish features, unambiguous. It had fins, also fish features. But those fins contained an internal skeletal structure that included a recognizable humerus, ulna, and radius, the three bones of the upper and lower arm, present in every tetrapod that has ever lived, including every human being alive today. The fins could not bear weight in the way limbs do, but they could push against a substrate. They could prop the front of the body up. They could, in the shallow, weedy, structurally complex environment of a delta swamp where normal swimming was impractical, provide purchase and purchase-derived movement in ways that conventional fins could not. Tiktaalik also had a neck, a genuine mobile neck, the first in the vertebrate lineage, allowing the head to move independently of the shoulders for the first time. It had ribs robust enough to support the weight of the thoracic cavity against the compression of gravity without the support of water. It had eyes positioned on top of a flattened skull that could scan above the waterline. It was, in the language of evolutionary biology, a mosaic. An organism carrying features from two different adaptive regimes simultaneously, straddling a boundary that had not yet been fully crossed. Acanthostega, from approximately 365 million years ago and discovered in Greenland, pushed the story further, and in a direction that initially surprised everyone who studied it. Here was an organism with genuine, unambiguous limbs, not fins modified in the direction of limbness, but limbs with digits, the structures that would eventually become fingers and toes in every subsequent tetrapod lineage. Acanthostega had 8 digits on each limb, a number that has since been found in other early tetrapods and suggests that the ancestral tetrapod digit count was variable before the 5-digit pattern became fixed. But Acanthostega also had gills. Its limbs, robust as they were, show skeletal features inconsistent with weight-bearing locomotion on land. The leading interpretation among paleontologists is that Acanthostega was primarily aquatic, that its limbs evolved not for walking on land but for moving through the dense, structurally complex vegetation of shallow aquatic environments, where fins were mechanically insufficient. Limbs, in other words, may have been an aquatic innovation first, a solution to the problem of navigating tangled underwater habitat before they became a solution to the problem of moving across dry ground. Ichthyostega, roughly contemporary with Acanthostega and also from Greenland, presents a different picture. Its skeletal anatomy is more consistent with terrestrial weight-bearing. Its ribs are overlapping and robust in a pattern that would have supported the thorax against gravitational compression. Its limbs are oriented in a way that could generate propulsive force against a solid substrate. But Ichthyostega was not a modern tetrapod in its locomotory style. Analysis of its skeletal geometry suggests it moved on land with a motion more reminiscent of a seal's crutching gait than the alternating limb swing of a salamander or lizard, pulling the front of the body forward with the forelimbs while the hindquarters followed. It was functional, it was real, confirmed locomotion on solid ground by a vertebrate. But it was costly, mechanically inefficient by the standards of what would eventually evolve, and probably constrained to short excursions rather than sustained terrestrial activity. The biomechanical challenges these organisms were navigating cannot be reduced to simple narrative. Gravity acts on every tissue, every organ, every fluid-filled cavity simultaneously. A fish lifted from water does not simply lose buoyancy. Its entire internal organization, evolved for an environment where weight is largely offset, is suddenly confronted with a force it has no mechanism to manage. The lungs, which in water are partly supported by the surrounding hydrostatic pressure, must now expand against the full weight of the thoracic wall. The heart must pump blood vertically against gravitational resistance in ways aquatic circulation does not require. The brain, cushioned in water-dense cerebrospinal fluid that reduces its effective weight dramatically, rests more heavily against its bony housing in air. These are not trivial adjustments. They are systemic challenges requiring coordinated anatomical and physiological modifications across multiple organ systems simultaneously. And yet the fossil record shows that they were met incrementally across a lineage that was being continuously shaped by the selection pressures of an environment that rewarded any solution to any one of them. Desiccation remained a persistent threat. The skin of early tetrapods was not the impermeable, keratinized integument of modern reptiles. It retained significant moisture permeability, meaning that time spent on land came with a continuous and physiologically significant cost in water loss. Reproduction presented perhaps the most fundamental constraint of all. Early tetrapods retained aquatic reproduction, returning to water to lay eggs in a medium where fertilization and embryonic development could proceed without the threat of desiccation. This reproductive tether to water would persist for tens of millions of years, defining the ecological range of amphibians to this day. The fully terrestrial life, the life independent of standing water for reproduction, would require another innovation that had not yet arrived. Now place yourself, if you can bear the conceptual distance, in the body of one of these creatures on the margin of a Devonian swamp, as the dry season tightens its grip on the landscape. The pool you inhabit has been shrinking for weeks. The oxygen in the water around you has fallen to levels your gills can barely process. You surface repeatedly, gulping air into your primitive lungs with a rhythm that has become as necessary as anything your gills have ever done. The water is warm and thick with decaying vegetation. Around you, other organisms are dying. Those that cannot breathe air, those whose physiology has no fallback when the dissolved oxygen disappears. But you are still here. You are still breathing. Your forelimbs press against the muddy bottom as you orient yourself toward the surface for another breath. And in that pressing, in that mechanical interaction between bone and earth, something is happening that will not be recognized as significant for 400 million years. The pool shrinks further. The mud at its margin dries and cracks under a sun that beats down through an atmosphere still different from our own. And at some point, not with drama, not with intention, not with anything that could be called decision, you move. Your forelimbs find purchase on the mud beyond the water's edge. Your body, heavy in a way it has never been in water, drags forward. Your lungs pull air. Your heart pushes blood through a circulatory system not fully prepared for this orientation. You have not evolved for this moment. You are surviving it anyway. You are our ancestor, and you have just changed the world. 50 million years is enough time to build a world. Not the cautious, experimental, marginal world of the Devonian shoreline, where vertebrates pulled themselves onto mud in isolated acts of physiological desperation, and arthropods navigated barren rock in the narrow band of moisture near the water's edge. But a genuine world, layered and complex and loud with biological activity from the ground to the upper canopy. By the time the Carboniferous period reached its full expression, roughly 300 to 359 million years ago, the land had undergone a transformation so complete and so dramatic that the Devonian pioneers who had made it possible would have been unrecognizable as ancestors of the creatures now filling it. What had been marginal was now central. What had been experimental was now the dominant theater of life on Earth. And what was happening in that theater, the scale of it, the density of it, the sheer biological extravagance of it, was something the planet had never produced before and would never quite produce again in the same form. The forests came first, and they deserve to be understood not as forests in any modern sense, but as something stranger and more alien than that word typically conjures. The dominant trees of the Carboniferous were not angiosperms or conifers or any group that survives today in recognizable form. They were Lycopsids, relatives of the small, low-growing club mosses that persist in modern forests as ground-covering plants rarely exceeding a few centimeters in height. In the Carboniferous, under atmospheric and climatic conditions that amplified biological growth to a degree without modern parallel, Lycopsids grew to heights of 30 to 40 meters, their trunks scaling upward in diamond-patterned bark that left fossil impressions so distinctive they remain immediately identifiable in the rock record. Lepidodendron and Sigillaria, the most abundant of these giant lycopsids, reproduced through spores rather than seeds, their life cycles tied to wet conditions in ways that reinforce the swampy, waterlogged nature of the lowland basins where they grew most densely. These were not isolated groves. These were continent-spanning forests stretching across the tropical belt of the Carboniferous world. A living, breathing, biologically active system of staggering biomass that covered areas measured in millions of square kilometers and drove atmospheric chemistry in ways that would reshape the conditions for everything living within it. The mechanism driving the most consequential atmospheric change was photosynthesis, operating at an unprecedented scale. All those lycopsid trees, all that fern understory, all the spreading botanical infrastructure of the Carboniferous forest was pulling carbon dioxide out of the atmosphere and fixing it into organic carbon at rates that the planet's geological carbon cycle could not fully compensate. When these trees died, in the waterlogged, anoxic swamp conditions that characterize the lowland basins, they did not fully decompose. The microbial communities capable of breaking down the complex polymer lignin, the structural compound that gave Carboniferous trees their extraordinary height and rigidity, had not yet evolved to process it efficiently at scale. Dead trees fell into stagnant water and accumulated layer upon layer across millions of years, becoming the coal deposits that would later define the period's name and fuel the Industrial Revolution. But the immediate consequence was atmospheric. Carbon was being removed from the active cycle and buried, and as carbon dioxide levels fell, oxygen levels rose. By the late Carboniferous, atmospheric oxygen concentration had reached levels estimated at 35% or higher, compared to the 21% that characterizes the modern atmosphere. And the consequences of that elevation were written into the bodies of every animal alive at the time. Oxygen is not simply a resource for animal physiology. At sufficient concentrations, it is a growth amplifier. The respiratory systems of arthropods, those book lungs and tracheal networks that had carried the first land pioneers across Silurian rock, function through passive diffusion of oxygen through the body's tissues. This system is physically constrained. The efficiency of diffusion falls off with distance, which means that in a modern 21% oxygen atmosphere, insect body sizes are limited by the depth to which oxygen can penetrate their tissues. Raise the atmospheric oxygen concentration dramatically, and that diffusion limit expands. Bodies that could not be adequately oxygenated at modern oxygen levels become physiologically viable. The result in the Carboniferous was gigantism on a scale that strains the imagination, even when confronted directly with the fossil evidence. Meganeura was a griffinfly, not technically a dragonfly in the modern taxonomic sense, but close enough in form and ecological role that the word dragonfly captures its nature better than any technical alternative. Its wingspan reached 70 centimeters, 70 centimeters of membranous wing, veined and translucent, attached to a body the length of a human forearm, navigating the airspace of the Carboniferous forest canopy with the compound-eyed precision of a predator that had no aerial competitors anywhere in its size range because nothing else of comparable scale had yet evolved powered flight. It was not alone in its gigantism. Arthropods, a relative of modern millipedes, reached body lengths of 2.5 meters, moving through the forest floor as the largest land invertebrate in Earth's history. Scorpions and early spider relatives reached dimensions that would be difficult to regard with equanimity at any distance. The oxygen-rich Carboniferous was not simply a more productive version of what had come before. It was a different world, operating at a different scale, where the bodies of arthropods had expanded into size ranges governed by physics that the modern atmosphere does not permit. The predator-prey dynamics this created were entirely without precedent. The forest floor was no longer the relatively simple environment of a detritivore consuming plant debris undisturbed. It was a complex, multi-layered arena of pursuit and evasion in which both hunter and hunted had been amplified to dimensions that made every interaction consequential. An Arthropods moving through leaf litter on the forest floor was doing so in an ecosystem that also contained amphibians of substantial size, early arachnid predators of considerable capability, and griffinflies overhead that could take vertebrate prey. The selective pressure generated by this complexity was not gentle. It was an evolutionary forge, and the animals being shaped by it were developing behavioral and anatomical sophistication at rates that would have been impossible in the simpler ecosystems of earlier periods. The amphibians, the vertebrate pioneers whose lineage traced back through Ichthyostega and Acanthostega to the lobe-finned fish of the Devonian swamp, had radiated into the Carboniferous forest with an enthusiasm that the ecological opportunity entirely justified. Temnospondyls, a diverse and enormously successful group of early amphibians, occupied body sizes ranging from small, agile forms moving through the understory to enormous, flat-headed predators exceeding 2 meters in length that ambushed prey from the margins of the ubiquitous swamp water. Ariops, one of the better-known temnospondyls from slightly later in the record, presents a body plan that communicates predatory intent with immediate clarity. A broad, heavily built skull armed with conical teeth, a robust body carried on substantial limbs, and a posture that positioned it effectively for the kind of sudden lunge that catches prey before flight response can be completed. These were not transitional forms still experimenting with terrestrial existence. They were fully committed land predators, occupying ecological roles that would be filled by crocodilians and large lizards in later periods, and doing so with considerable biological success. But amphibians, for all their carboniferous abundance, carried a constraint within their biology that had never been fully resolved from the days of their Devonian ancestors. They were tied to water, not for their own survival. Many could range considerable distances from standing water when conditions permitted, but for reproduction. Amphibian eggs are permeable structures without a waterproof outer membrane, which means they desiccate fatally if laid in air. Fertilization is typically external, requiring the aquatic medium to bring gametes together. every amphibian alive today and every amphibian that moved through the Carboniferous forest, regardless of how large or how formidable, had to return to water to reproduce. This was not a minor logistical inconvenience. It was a fundamental ecological boundary defining the geographic limits of every amphibian population that ever existed, keeping them within reach of standing water, preventing them from colonizing the drier interiors of continental landmasses, constraining their evolutionary possibilities in ways that would ultimately determine their place in a world increasingly dominated by organisms that had found a different answer to the reproductive problem. That answer was the amniotic egg. It is difficult to name a single evolutionary innovation in the history of vertebrate life on land that rivals the amniotic egg in its consequences, because the amniotic egg did not merely solve a problem. It dissolved a boundary that had constrained vertebrate distribution and ecological possibility for tens of millions of years. The amniotic egg is, in essence, a private pond carried inside a shell. Its defining features are a set of embryonic membranes, the amnion, chorion, and allantois, that surround the developing embryo with a fluid-filled environment, manage gas exchange through a permeable but water-retaining outer structure, and store metabolic waste in a dedicated membrane so it does not contaminate the embryonic environment. The result is an egg that can be laid in air, in dry substrate, far from any standing body of water, and still provide the developing embryo with everything it needs for the full duration of its development. The first amniotes, the lineage from which all reptiles, birds, and mammals are descended, appeared in the early Carboniferous, approximately 312 million years ago. Hylonomous laeli, preserved in fossilized tree stumps in the coal swamp deposits of Nova Scotia, represents one of the earliest confirmed amniotes in the fossil record. A small, lizard-like creature that would not have attracted particular attention in the Carboniferous forest, easily overlooked against the spectacle of giant arthropods and massive amphibian predators sharing its ecosystem. But Hylonomous and the lineage it represented carried within its reproductive biology something that would ultimately reshape the vertebrate world more thoroughly than any anatomical feature visible in its fossilized skeleton. It did not need water to reproduce. And that fact, which would have carried no immediate competitive advantage in the waterlogged lowlands of the Carboniferous coal swamp, was a key that fit a lock the world had not yet presented. The dry continental interior, the elevated terrain beyond the swamp margins, the vast expanses of land that amphibians, for all their evolutionary success, could never permanently colonize. It is worth pausing at the scale of what the Carboniferous represents in the context of this longer story. The creatures that had crawled onto land in the Silurian, small, segmented, moving cautiously across damp rock near the water's edge, had been the seed of something that 50 million years of evolutionary elaboration later had become a full planetary ecosystem of staggering complexity and biomass. Land was no longer a hostile frontier that organisms visited under duress. It was the dominant theater of biological innovation, the place where the most consequential evolutionary experiments of the era were being conducted, the environment whose conditions were driving the most rapid diversification of body plans, behaviors, and ecological strategies in the history of complex life. The ocean, which had seemed so irreplaceably rich and stable to any organism contemplating the barren Cambrian shore, had not diminished. But it had been joined by something that rivaled it in biological complexity and surpassed it in evolutionary dynamism. And yet, the Carboniferous world, for all its spectacle, was not stable. The same biological processes driving its extraordinary productivity were simultaneously undermining the atmospheric conditions that made its most extravagant features possible. Oxygen levels that had climbed to their Carboniferous peaks were not going to remain there indefinitely. The forest fires that ignited with terrifying ease in the oxygen-saturated air, burning with an intensity and spread that no modern fire can replicate, leaving charcoal horizons preserved in the coal beds that now yield this information, were themselves a feedback mechanism, consuming vegetation and releasing stored carbon back into atmospheric circulation. The climate was shifting as the Carboniferous progressed, the vast tropical forests fragmenting as the continental configurations of Gondwana drove glaciation across the southern landmass. The coal swamps that had sustained the Lycopsid giants were drying and contracting, and in their contraction they were eliminating the waterlogged habitats that amphibian reproduction required, while leaving intact the drier environments where amniotic reproduction offered its clearest competitive advantage. Dominance in the history of life on Earth is always provisional. The Carboniferous had demonstrated beyond any possibility of doubt that land could sustain ecosystems of genuine grandeur. But the conditions producing that particular grandeur were already changing. The giants of the oxygen-rich forest, the griffin flies, the giant millipedes, the massive amphibian predators of the coal swamp margins, were already approaching the end of the specific atmospheric window that had made them possible. Something leaner, more independent, more adaptable to a world with less reliable water and lower oxygen was already moving quietly through the forest understory. It was small, it looked unremarkable. It would inherit everything. The Carboniferous did not end gently. It ended the way most geological periods end, through the slow accumulation of changes too gradual for any living organism to perceive, too vast in their consequences for any ecosystem to fully absorb. The coal swamps contracted. The vast Lycopside forests that had defined the lowland basins for 60 million years fragmented into isolated refugia, their waterlogged substrates drying as the climate shifted, and the continental configuration of Pangaea drew moisture away from the interior of a landmass that had become the largest single piece of continuous terrain the planet had assembled in hundreds of millions of years. The atmospheric oxygen that had powered Carboniferous gigantism began its long descent back toward levels the modern world would recognize. The griffin flies vanished. The great amphibian predators of the swamp margins found their breeding habitats shrinking, their populations fragmenting, their ecological dominance eroding in the face of conditions their reproductive biology had never been built to withstand. What replaced them moved quietly, efficiently, and with a biological independence that the drying Permian world rewarded with a thoroughness that nothing dependent on standing water could match. The reptiles did not conquer the Permian through spectacular anatomical innovation or sudden ecological dominance. They expanded into it incrementally, the way evolutionary success almost always actually works. Through the patient accumulation of marginal advantages across thousands of generations, each generation slightly better suited to the specific conditions of a world that was slowly but definitively tilting in their favor. The amniotic egg, that private pond carried inside a shell, was the foundation of everything. It meant that dry upland environments, elevated terrain beyond the reach of seasonal flooding, the vast continental interiors that had never sustained vertebrate populations for more than transient visits. All of it was now accessible for permanent colonization. Where amphibians could only project their populations outward from water bodies like light from a fixed source, Amniotes could go anywhere that food and temperature permitted. And in the Permian world, that difference in reproductive freedom translated directly into geographic range, population size, ecological diversity, and evolutionary opportunity on a scale that no amphibian lineage could compete with indefinitely. Among the reptiles radiating through the Permian, one group deserves particular attention, not because it was the largest or the most ecologically dominant in the short term, but because it was ancestral to something that would eventually include the reader of this story. The synapsids were, by external appearance, not obviously distinct from other early reptiles of the Permian. They were characterized by a single opening in the skull behind each eye socket. The temporal fenestra, a structural modification that allowed for the attachment of larger, more complex jaw musculature than the solid-skulled reptiles possessed. This seemingly minor anatomical detail was the visible surface of a deeper reorganization of the synapsid body plan that, over tens of millions of years, would produce increasingly sophisticated jaw mechanics, increasingly differentiated tooth forms, increasingly complex metabolic and thermoregulatory systems, and ultimately the capacity for endothermy, the internal generation of body heat that defines mammals today and that carries with it a set of ecological capabilities that ectothermic animals simply cannot match. The early synapsids, pelycosaurs like Dimetrodon, with its dramatic dorsal sail that almost certainly functioned in thermoregulation, were the dominant large-bodied terrestrial vertebrates of the early Permian. They occupied the apex predator and large herbivore niches across Pangaean landscapes with a comprehensiveness that suggests genuine ecological command. But Dimetrodon and its relatives were themselves only an early chapter in the synapsid story. The therapsids that followed, more derived synapsids with increasingly mammal-like skeletal organization, increasingly upright postures, and increasingly complex metabolic physiology, carried the lineage forward through the catastrophic Permian-Triassic extinction event, the most severe mass extinction in Earth's history, during which an estimated 96% of marine species and 70% of terrestrial vertebrate species were eliminated. The therapsids did not escape that catastrophe unscathed. They were devastated by it, but they were not eliminated. And in the aftermath of the Permian extinction, in the ecological vacuum left by the collapse of the world's most complex terrestrial ecosystems, their survivors found themselves in a landscape of opportunity as radical as anything their distant ancestors had encountered on the shores of the Devonian swamp. The Triassic recovery was complicated by the rapid diversification of the archosaurs, the lineage that would produce the crocodilians, the pterosaurs, and the dinosaurs. And for a period, the synapsid lineage was ecologically marginalized, reduced in body size and ecological scope by competition with archosaurian forms that proved devastatingly effective in the warm, seasonally arid environments of the Triassic world. The mammals that emerged from surviving cynodont therapsids during the Triassic were small, likely nocturnal, metabolically sophisticated in ways their body size required, and occupying the ecological margins of a world whose large-bodied niches had been claimed by creatures that would dominate terrestrial ecosystems for the next 150 million years. The age of dinosaurs was not a period of mammalian flourishing. It was a period of mammalian endurance, of surviving in the shadows, maintaining lineages, accumulating the genetic variation and metabolic refinements that would eventually be expressed on a global scale when the conditions that had suppressed mammalian expansion were finally catastrophically removed. That removal came 66 million years ago in an event whose violence exceeds easy description. The Chicxulub impactor, an asteroid or comet approximately 10 kilometers in diameter, struck what is now the Yucatan Peninsula of Mexico with a force that released energy equivalent to billions of nuclear weapons simultaneously, triggering wildfires across continents, injecting sufficient material into the upper atmosphere to block photosynthesis globally for months to years, and collapsing the food chains that sustain the non-avian dinosaurs with the speed and totality that geological time rarely produces. The mammals that survived were not the largest or the most ecologically dominant creatures of the Cretaceous. They were the small, the generalized, the metabolically flexible. Animals whose endothermy allowed them to function in the cold and dark of the impact winter, whose omnivorous tendencies gave them access to food sources that persisted when the primary productivity of large plant communities crashed, whose reproductive biology in many lineages had moved beyond even the amniotic egg toward internal gestation that protected developing young from environmental instability. They survived not because they were superior to what was lost, but because the specific nature of the catastrophe intersected with their specific biology in ways that proved, by the narrowest of margins, compatible with continuation. What followed, the Paleogene radiation of mammals into the ecological space vacated by the non-avian dinosaurs, was an evolutionary expansion of extraordinary speed and diversity. Within 10 million years of the Cretaceous extinction, mammals had radiated into large herbivore niches, apex predator roles, arboreal lifestyles, semi-aquatic environments, and aerial locomotion. Within 20 million years, the basic outlines of most modern mammalian orders were established. The continents, drifting across the planet's surface in their slow tectonic procession, were carrying these diversifying mammalian lineages into geographic isolation from one another, accelerating speciation and producing the patchwork of regional faunas that would eventually be encountered by the one lineage whose subsequent trajectory no reasonable observer of the early Paleogene could possibly have predicted. The primates that gave rise to the human lineage were unremarkable members of an unremarkable mammalian order for most of their evolutionary history. Arboreal creatures of the tropical forest canopy, their grasping hands and forward-facing eyes refined for a life in three-dimensional tree structure rather than for anything that would follow. The specific sequence of events that led from the last common ancestor of chimpanzees and humans to the emergence of bipedal, large-brained hominins on the African savanna involved a combination of climatic change, habitat fragmentation, and selective pressures whose precise reconstruction remains one of paleontology's most actively debated questions. But the outcome is not in doubt. Somewhere between 6 and 7 million years ago, a lineage began that would eventually produce a creature capable of standing upright on two limbs, freeing its forelimbs for manipulation, expanding its neural architecture beyond any previous primate scale, and developing the capacity for the kind of cumulative, transmitted symbolic knowledge that eventually allowed one species to look back across 500 million years of evolutionary history and ask how it came to be standing where it was standing. That question, how we came to be here, on land, walking upright, capable of asking anything at all, dissolves into an answer that is not a single moment, but an unbroken chain of contingent decisions made by organisms that had no capacity for decision, across a span of time that defeats any attempt at genuine comprehension. Every step a human being takes on solid ground is the latest expression of a biomechanical inheritance that began in the muddy margins of a Devonian swamp, was refined across the Carboniferous forest floor in the drying Permian plains, was tested by catastrophes that eliminated 90% or more of all life on Earth, not once but repeatedly, and emerged from each test not because of inevitability but because of the improbable contingent survival of enough individuals carrying enough genetic variation to continue. There is no guarantee in this story. There is no direction, no trajectory aimed at any particular outcome, no force guiding evolution toward complexity or intelligence or anything resembling purpose. There is only survival and variation and time, and the extraordinary fact that the combination of those three things, operating across scales that dwarf human experience in every dimension, produced a world in which something can look back and recognize the shape of what happened. The land was empty once. Not metaphorically empty, not sparsely populated, a genuinely, absolutely, biologically empty in any meaningful sense. Rock and radiation and silence extending across continents that had never been touched by anything with a nervous system. Anything that moved with coordinated intent. Anything that left a mark more complex than a chemical trace in ancient stone. That emptiness was not a waiting state. It was not pregnant with inevitability. It was simply empty, and the processes that filled it were not aimed at filling it. They were aimed at nothing. They were the blind operation of chemistry and physics and the differential survival of variance in changing environments, playing out across timescales that no mind evolved to think in years or decades, or even centuries can properly hold. And yet, something moved in the shallows. Something pressed its weight into ancient mud and left an impression that the stone preserved. Something pulled itself onto land that had never felt the weight of a vertebrate body, breathed air that had never passed through vertebrate lungs, and continued existing in a place that had every reason to kill it. Not because it chose to, not because it knew what was at stake, but because it could, barely, and because barely, in the mathematics of evolution, is always enough to change everything. Every footprint you have ever left in soft ground is that first impression, carried forward through 500 million years of survival against odds that were never calculated and never needed to be. The chain is unbroken. The land that was empty is yours. And it began as all things of consequence begin, with something small moving carefully into the unknown.